So welcome everybody to our last this year webinar on innovation in neuroscience. And my name is Dovilja Kurmanovic-Ute. I'm innovation agent and doctoral candidate at Halvo University, and I'm a part of organizers for this webinar. And today's topic is From Research to Business, same name as Business Finland from Inco. For this reason, we also have Helsinki Innovation Services today with us. Thanks, Kaisa, for joining. In this webinar, we aim to show you examples and share experience on the innovation path from lab research to market. And without further ado, I briefly introduce our first speaker, researcher and innovator Timo Myöhänen. Timo is a group leader in Neuroprep project at the University of Helsinki. He holds the title of docent at Division of Faculty of Pharmacy. Timo is also assistant professor at Faculty of Pharmacy, senior researcher at Division of Pharmacology and Pharmacotherapy, principal investigator at Regenerative Pharmacology Group, and also supervisor for doctoral program in drug research. Welcome, Timo. And now screen is yours.
Okay, thank you very much for the kind introduction and invitation for this Innovation in Neuroscience seminar. So I'm going to present a little bit about our Neuroprep project. As you can see, this Neuroprep has very ambitious aims. It's about stopping the Alzheimer's and Parkinson's disease. And I'm going to introduce you a little bit how we aim to get here. And as maybe many of you know, neurodegenerative diseases are quite large set of diseases. Parkinson's disease has approximately 10 million patients worldwide. Alzheimer's, then again, way bigger with 50 million patients. And common for all of these is that there is a neuronal degeneration in some part of the brain, causing clinical symptoms that then can be movement disorders or dementia. And another common thing for these is that there is no curative treatment for neurodegenerative diseases. So current treatments, they can only relieve the symptoms, but they cannot really block or even delay the neural degeneration that then continues all the time in the brain, causing all the time more severe symptoms. And what's important is that this amount of these diseases, amount of the patients with these diseases is basically constantly increasing. In the Western countries, the aging has been already slightly flattened, but then again, there are these former third world countries like China, where the aging has started to increase. And we have to remember that the age is the most significant risk factor for these diseases. So basically, aging increases the risk, and therefore, when there's more aged people, there will be more cases with the neurodegenerative diseases. And since we are speaking about the Business Finland project, we always need to remember a little bit the market, and it's also an increasing market. Already, with the current therapies that are not curative, the market is estimated to be $9 billion, and it's been estimated that it elevates to $12 billion in 2024. So it's clear that we are in the need of new treatments that would be able to at least reduce the disease progression or even block them, which would be, of course, the optimal situation. And our approach has been PrEP, oligopeptidase, but let's just say it as a PrEP. And it's a serine protease. It cleaves short peptides. And back in the old days, they noticed that there is changes in PrEP enzyme activity during the aging and in the certain neurodegenerative diseases. And this served as a background to develop small molecule PrEP inhibitors. And this drug development was quite intense, let's say from 90s to 2005. And for example, our reference compound KIP2047 was developed by that time. And the idea was that if we block the enzymatic activity of PrEP, we are able to elevate neuropeptide levels in the brain and enhance memory. The idea was good. Preclinical models showed some effect, good enough that a couple of the PrEP inhibitors were tested in the clinical trials. But there wasn't any effect. And looking at back now, getting a memory effect on aged, healthy individuals is probably very difficult no matter what you try. But the important part was that PrEP inhibition was shown to be safe in phase 1 and 2 clinical trials. 3 of them were tested. KIP2047 wasn't one of them, but they were turned out to be safe. But when the clinical trials failed, of course, the interest in PrEP slightly decreased. Until 2008, a group from Belgium, they showed that PrEP can enhance the aggregation of alpha-synuclein, which is basically very easy to get in an in vitro setting, what happened here. But important was that by using PrEP inhibitors, they were able to block this effect. And why this alpha-synuclein is then again so important? So alpha-synuclein is one of the main components of Lewy bodies that are then again used as a hallmark for Parkinson's disease and a couple of other diseases called synucleinopathies. So the alpha-synuclein, when it finally forms the Lewy body, it's in this very tightly aggregated form. And recent studies have shown that it's actually something that happens in between this, when alpha-synuclein starts to aggregate and before it forms a Lewy body. That's toxic for the cells. And that's something where we've been able to target with the PrEP inhibitors. So there's about 10 years of science behind this, and this 15 minutes is not enough to go through that. But what we noticed is that PrEP makes a direct interaction with alpha-synuclein. It enhances its aggregation, and it also regulates autophagy, which is kind of a cellular recycle center that then again degrades these synuclein oligomers, aggregates, and other toxic species. And both of these are not related with this enzymatic effects of PrEP, but it's a protein-protein interaction. And in the best case, what we've been able to do in the animal studies, we showed that when we give an animal an overdose of alpha-synuclein by using virus vector, it starts to generate behavioral deficit. And when we start the treatment, when there is already a behavioral deficit, we are able to reduce or actually almost completely remove this behavioral problem and reduce these synuclein oligomers. And of course, this was very nice finding. But then again, we started to think that, well, maybe we need to now do something else with the PrEP inhibitors. And that's when we actually started to plan and apply this Business Finland projects. Because these former PrEP inhibitors like KIP2047, they are very, very widely patented back in the old days. And then there are even some new patents where they cover usually a lot of neurodegenerative diseases. The other thing, what we found in our assays, that this IC50 that we used to show that how effective these PrEP compounds, PrEP inhibitors are to inhibit the enzymatic activity of PrEP, it doesn't correlate with, for example, synuclein aggregation or autophagy induction. There were some very potent PrEP inhibitors that didn't do anything, and then some that were considered not potent at all in the micromolar range, and they were actually quite effective. And this served as an idea for us to start developing new PrEP ligands and initiate this project. And during this Neuroprep Business Finland project, we have developed 5 different series of new PrEP inhibitors, or I would say PrEP ligands. And as you can see, this red arrow is the KIP2047. And for example, this HOOP46, this blue arrow, they are way more effective in synuclein aggregation or to block the synuclein aggregation and to induce autophagy. And we've been doing this cellular screenings and eventually progressed to the animal models with these. And it was quite interesting to see that when we used or basically repeated this virus vector study that I showed earlier, but we used an alpha-synuclein vector that was more toxic than the one we used earlier. KIP2047 couldn't anymore block the behavioral deficit, but our new compound HOOP46 was able to do it. And it turned out to be a bit more effective or reducing synuclein. And it showed, both of them showed some neuroprotection, but it appeared that HOOP46 was a bit more effective also on this one. We are still under the studies, so therefore these are quite preliminary still. But it seems that these new compounds that were pretty good in the cell models, they were also effective in the animal models. And I was speaking about the Alzheimer's disease as well at the topic, though I didn't show any data about that. Hopefully we have pretty soon. I hope it will be as a Christmas present for me that we get the good results from the Alzheimer mouse model. But so far, there isn't any results from that. But we have to remember that there are several common toxic mechanisms among the neurodegenerative diseases. For example, cellular stress, neuronal overactivity, cell-to-cell spreading, and this toxic protein accumulation. And we know that with PrEP inhibitor, we can target on several of those. And that's why we think that the PrEP inhibitors can be applicable for several neurodegenerative diseases. We do have some data on the Alzheimer's cellular models, but I didn't present them at this time. So that's why we kind of think that our approach, this multi-targeting approach, might be successful if you compare it to the former approach where you target generally on one toxic mechanism at the time. Okay, that's all I had to say about this. Of course, acknowledgements, there is a lot of science behind this before even this Neuroprep project. And I'm happy to answer your questions.
Thank you very much, Timo, for a great presentation. And now let me introduce our next speaker, who is an enthusiastic innovator and researcher with entrepreneurial mindset, Marko Halvo, who will tell us about imaging MRI project at Aalto University. Marko is doctoral candidate in MRI research group at the Department of Neuroscience and Biomedical Engineering at Aalto University. And thanks, Marko, for coming. Now, screen is yours.
Thank you, Davile. Right. Like Davile said, I'm from the MRI brain imaging group at Aalto University. And I will first tell a bit about the project and then move on to challenges and experiences that we've gained during the project. And also what makes a successful commercialization project in neuroscience, in my opinion. But first, worldwide, 1 out of 6 people suffer from some kind of brain disorder. And in Europe, the annual cost for society is over 800 billion euros. Functional brain imaging is a tool used in diagnosis and treatment of many of them. However, today, imaging the electrical activity in the brain requires 2 studies. First, we need functional data that comes from magnetoencephalography or electroencephalography. But in addition to that, we need anatomical or structural data that usually comes from magnetic resonance imaging. The necessity of 2 studies makes this process inaccurate, impractical, and costly. So our value proposition is a hybrid brain scanner that can get both the functional and the structural data in a single scan. This improves the workflow and saves a lot of time. It also means that we can totally eliminate the worst case localization errors. And in addition, our technology enables completely new kinds of brain research. It has, for example, a unique contrast for cancerous brain tissue. The technology has been developed for over a decade. In the first EU-funded project, MEG-MRI, a scientific proof of concept was built. It had sensors at the back of the head. Then there was another EU-funded project, a FET project, Future in Emerging Technologies, in the Horizon 2020 program, called BreakBrain, and that culminated in a whole head research prototype. So now we have world's first prototype, which is able to measure whole head at once. Right now, we're further refining the prototype, and the clinical validation will start next year. We have two ongoing commercialization projects. Another EU-funded project, MaxPass, is a FET Innovation Launchpad project, and then we have the Business Finland-funded Tutli project. One patent for the pre-visualization method was granted in 2017, and we have two more applications in the PCT phase. The clinical validation studies that we have planned include patients with epilepsy, Alzheimer's disease, and brain cancer. In the case of epilepsy, we seek to decrease the time it takes for a clinician to analyze the data. And in brain cancer, early detection could save lives. And it's been estimated that spotting Alzheimer's disease early could save almost $8 trillion during the lifetime of the current population in the United States alone. This is our team in the Tutli project. Koos Evenhoven is the research group leader. He's been developing the technology since the beginning and is the world's topmost expert in ultra-low field MRI, instrumentation, and physics. Professor Risto Ilmaniemi is the head of our department. He's the father of the project. He's had a great track record in converting groundbreaking research into businesses. He's a pioneer in MEG, transcranial magnetic stimulation. And he also served as the CEO of NextStim in the beginning. Thomas Lemström is an expert in strategy and roadmaps, and I'm a product developer with entrepreneurial background. I joined the project 4 years ago while the BreakBrain project was ongoing. It had, I think, started 1 year before I started. Brain imaging is one of the fastest growing imaging segments with 7 to 9% growth per year. And we estimate that the immediate market for MEG-MRI will be 150 million by 2029. In addition, there is potential for another 350 million if findings support the use in all key application areas. Our competition comes mainly from the existing functional imaging modalities. They all have their shortcomings, but they also have a large installed base. Here you can see comparison of some key metrics between the existing functional imaging modalities and our technology. The unit cost for a scanner will be in the range from 2 million to 4 million euros. And we're going to the market in 3 phases. First, we're going to target research institutes because that's where we can immediately bring new value. Also, getting approvals from FDA and CE, or the FDA and CE approval can take time and then also may require clinical studies. So hospitals will come later. First, the research-oriented university hospitals, and once the clinical value is proven, the other hospitals. So that's what I have to say about the project. And then I'll tell a bit about the experiences we've had with the Tutli project and also Spark Finland program. So why apply for research to business funding? Well, first of all, I really like the application process. It's smooth, and it actually improves your project because it's a bit different from what they usually are. You're supposed to first pitch the idea, and then if you get a green light, then you can send in the actual application or the project plan. So it requires you to think of the impact of your projects and the alternatives for it. At least in Aalto, I think in Helsinki University as well, maybe in all universities, the innovation services organize the pitching workshop, so you can prepare for this. And I would actually suggest that you start from really, really short pitch, say 1 minute pitch, to really crystallize what's important in the project, and then you can add more stuff later on. Second of all, it's full funding in the sense that you can. It's substantial enough for you to actually complete the commercialization project. Many of these fundings you get are fairly small, so it's not. You won't be able to go through a full commercialization project with that funding only. But with this one, you can actually plan a project that will take you as far as to the market if your field is all right for that, and you're able to get enough funding for that. The amount of funding you get from Business Finland is 70%, so you still need to have some on top of that, but the scale is right. We're also Sparkies, so we're members of the Spark Finland community, and the number one reason we joined that was networking with advisors, investors, potential customers. We joined Spark Finland 1 year ago, and at that point we were still looking for a startup professional to join the steering group of our Tutli project. And through Spark Finland, we got a great additional member who not only is a startup professional, but he used to be a country manager for a large medtech company in Finland, and is familiar with all the problems we face as a newcomer trying to get into a market where these big players are already have established their position there. And in addition to this networking, constant feedback and benchmarking is a very valuable asset you get from Spark Finland. And as an extra, we've also benefited from training, for example, in regulatory matters. So for all neuroscience projects that apply for Tutli funding, I also recommend Spark Finland. So what can I offer as a tip for success? First, let's look at the challenges we've had. First of all, technology. In our case, in other cases, it might be like scientific challenges, but in our case, the technology is challenging mainly because the difference between the smallest signals we're trying to measure and the pre-polarization pulses we use is 14 orders of magnitude, which is huge in any field of engineering. Also, resources, both financial and human. So financial resources, especially after Tutli, like I said, the Tutli funding is substantial, so it covers pretty much everything we need at this point. But apart from planning the project, your number one concern during the Tutli should be covering the funding for after the Tutli ends. or research to business, as it's called these days. Especially in capital-intensive fields like ours, or projects with low technology readiness level. It might not be so easy to, or after the Tutli project, you won't be ready to enter the market yet. You still need some additional funding. And even in the case you have some kind of easy or relatively easy projects, for example, an app, you still need some funding, be it venture capital or whatever it is, but you still need to get some funding to get to the market. One way to succeed is need-based innovation. So you identify a need and then develop a solution to the underlying problem. But since we're all researchers, then this may not be sort of the path to take. You already have a scientific or technological challenge that you wish to solve, and that's how our project also started. Combining two inherently incompatible technologies with the challenge that they wanted to overcome. And that's been the focus. But since the beginning, there has been a focus on the impact, focus on the value, and that's what I encourage you to do. So think about the value and impact. There are tools like, for example, Lean Canvas that you can use to design the value proposition, and I recommend those. They also usually improve the research project. Like I said, in pitching, you need to think about the impact and alternatives. And once you go through this process, it usually also gives ideas for research. And if nothing else, they improve your chances to get also scientific funding. And as last point, I would like to say that team is the most important thing in any commercialization project. So try to get the people you need. People with money or people who are smarter than you are good members of the team. And a bit short-staffed on the engineering side at the moment. So all that, when I showed the team photo, everyone we got is in that photo, and we would need some more people. So if you know talented people who would like to join an imaging project, then I'd be happy to know. Thank you. I'll be happy to answer any questions you might have.
Thank you, Marco. So let's go to our next speaker, Philip Sheparian, who is an innovator and a medical doctor. He obtained his medical degree in Düsseldorf University, Germany. So Philip holds also docent title and focus on microbiome role in Parkinson's disease. And as you may guess, today he will tell us about microbiome-based diagnostics for Parkinson's disease. So thank you, Philip, for giving us a great opportunity to learn about your project.
Okay, thank you very much for the invitation. All right, so my presentation is rather short because I just want to leave also enough time for discussion. So my story is a bit different from that of the previous speakers in the sense that so this project was not was not supported by Tutli because it is not related to at this stage anymore to the university, and so the Tutli projects are require that the university has a stake in the project, and so in this case for several reasons it's not the case. So in that sense, the whole process of funding and and progress is a bit different in this case. So maybe even though Timo already gave a very nice introduction, I also have a few main points about Parkinson's disease. So Parkinson's disease is my main focus clinically and also in research. And it's the second most common neurodegenerative disorder. And the main symptoms, as Timo mentioned, is a movement disorder syndrome, so slowness of movement, muscle stiffness, tremor, and in the advanced stages also posture instability, so problems with balance. And we have about 15,000 patients in Finland, and so Timo mentioned 10 million patients. In my source, it was mentioned 5 million patients, so probably the truth is somewhere in between. So there's many million patients in the world, and there are several problems related to the treatment of Parkinson's disease. First of all, we diagnose Parkinson's quite late, so in the stage when the patients exhibit motor symptoms. We know that already about 60% of the dopamine-producing nerve cells in the brain have died. We know that this correlates with the motor symptoms, and obviously in terms of stopping or slowing down a disease such as Parkinson's disease, we would like to treat as early as possible. So we're not very well in picking Parkinson's disease up currently. And we don't know what is causing Parkinson's disease, but we know that it is chronically progressing and that nerve cells progressively die, but we don't know exactly why this is the case. And I think what is the most striking thing about our current treatment of Parkinson's disease is that the best drug, the most effective drug that we're using for Parkinson's disease, which is named levodopa, it has been discovered in 1967, so it was before the first man landed on the moon, and it is still the most effective treatment for Parkinson's disease, and no really any major revolutions have happened in the pharmacological non-invasive treatment of Parkinson's disease. We have learned quite a lot in recent years about the how and where Parkinson's disease starts. And especially it has been recognized that the gut seems to play an important role in Parkinson's disease. Recently, we have even seen reports that there may be a certain subgroup of Parkinson's patients where the disease actually starts in the gut and not in the brain, but then the pathology would spread from the gut upwards to the brainstem, and only then we would see the classical motor symptoms. And so there is quite strong evidence that there is widespread dysfunction in the gut, and there is a low-grade inflammation in the intestines, and that we can also see the alpha-synuclein inclusions that Timo mentioned already in his talk, that we can see the same neuropathological changes also in the gut. And there's, even though all mechanisms are not known, but we know that there is neuroinflammation going on, which is probably progressively also or is related to the disease progression, mitochondrial dysfunction, and oxidative stress. So and that is, I think, also one problem of the whole Parkinson's disease field is that we still have a lack of understanding how these manufacturers really interact and contribute. And that is why we got interested in the gut-brain axis and its role in Parkinson's disease. So just to quickly get you acquainted with the concept of the gut-brain axis, basically it means that we have learned how much the gut, and in particular gut bacteria, are interacting with the brain, for example, by programming the immune system, but also by directly affecting the vagus nerve, and also by producing neurotransmitters and other metabolites such as short-chain fatty acids that affect brain function and also structural integrity. And so our interest is in finding out whether we could develop a completely new way of tackling Parkinson's disease by using the gut-brain axis to influence the pathological process of Parkinson's disease. But in that in that context, we and all others that are trying to develop treatments for Parkinson's disease, we're faced with the problem that there is a strong heterogeneity in the symptoms, but probably also in the pathology between the mechanisms that are involved in Parkinson's disease between different patients. And so at this stage, we don't have any tool that we could use to specifically identify certain subgroups of patients and could try to develop specific treatments for these subgroups based on their underlying pathology. And I think that one of the main reasons why so far all efforts to slow down Parkinson's disease have failed is that usually these treatments or these drugs are being tested in a group of patients that probably consists of different subgroups, where you could have certain individuals that have a very strong response to the specific treatment, but you could also have a reasonably large group that doesn't respond at all because they have a different underlying mechanism. And so the same seems to be true also for the involvement of the gut in Parkinson's disease. And so we desperately need biomarkers that we can use to identify those patients that are the most suitable for the treatment concept that we would like to test. And so we started studying the gut microbiota in Parkinson's disease in 2012. And we were fortunate in the sense that we were the first to be able to publish about these findings. And since we were the first, we were also, we had the chance to try to get patents for certain applications for microbiome analysis in the diagnosis or diagnostic process of Parkinson's disease, and also related to certain treatment aspects. So here I will focus on the diagnostic side of it. And so what we found was basically a reduction in the abundance of Prevotella bacteria in the stool of Parkinson's patients, and an increase of Akkermansia. We also found some other bacteria that were altered, but the strongest signal was this contrast between Prevotella and Akkermansia. And we were also quite fortunate because those findings eventually turned out to be the best reproduced findings across different cohorts and continents and ethnic groups to be altered in Parkinson's disease. And so it seemed it was the most robust finding as well. And based on literature and subsequent research, there is some evidence for these bacteria being related to the integrity of the mucus barrier of the gut. So basically protecting the especially Prevotella, protecting the gut and the human organism from, for example, toxins and other pro-inflammatory factors that can be in the gut lumen. And also they seem to be related to the production of short-chain fatty acids that also regulate the immune system, and that may even affect neuroinflammation in the brain. And so with respect to the application of potential microbiome modifying treatments, we think that it is crucial to identify those patients that have the highest chances of benefiting from such a treatment. And it turned out that what you see on the right side in the graph is that actually when you look at the whole population of Parkinson's patients, and there's only about 25% of patients that actually have a clearly abnormal microbiome, whereas the rest is basically, you cannot really discern them from healthy control subjects in terms of their microbiome. So this, again, emphasizes my previous point that we need to find markers to identify such subgroups, and that's what we basically did. So we took our data from our research and our studies on the gut microbiome of Parkinson's disease, and developed a test platform that allows us, with a turnaround of about 1 to 2 weeks, to identify from a fecal sample of a Parkinson's patient whether this patient has an abnormal microbiome in comparison to other Parkinson's patients and healthy controls. And those would be the patients you see on the right side. Those large black dots would be those so-called dysbiotic patients, so that have an abnormal microbiome. And so for the concept of using microbiome analysis in the diagnostics of Parkinson's disease, we have granted patents in Europe and the US, but we also have undisclosed proprietary algorithms that we use to analyze this data and to perform this testing. And so what we're doing in the next step is and what we have received funding for is we will start recruiting. Actually, we have started recruiting patients now. So we will conduct a clinical trial in 3 centers in Finland, where we will recruit 48 Parkinson's patients. And we will use the test that we developed to identify only those patients that have an abnormal microbiome. And so we will restrict the study to people that have the abnormal microbiome. And when we in these 48 patients, we will give them what is called a fecal microbiota transplantation. So essentially replacing their abnormal microbiome with that of a healthy donor. And we will then follow up the safety of the procedure and the functional efficacy, so how it potentially alleviates Parkinson's symptoms, and we will do also a large array of target engagement studies. And so we hope that this will help us to establish this test platform as an easy and clinically feasible tool to identify a subgroup of Parkinson's patients that is most likely to benefit from microbiome modulation treatment. And yeah, this was actually my last slide, and I'm happy to further discuss on this topic or to answer any questions. Thank you very much.
Okay, thank you very much, Philip. And lastly, we will have guys telling us about Business Finland call.
Yes, okay. Good afternoon, everybody. I'm quite sure after these inspiring talks by the gentlemen, hopefully quite many of you have gotten interested in probably commercializing some of your own ideas or inventions. This is actually the perfect timing for that. The research to business funding tool has 2 funding calls annually, and the next one is in March next year. But before getting there are some critical steps still to pass. And that's why actually it is good to activate yourself now if you're interested in this funding type. The advice I will be sharing here are for the University of Helsinki researchers. They will be sort of detailed of the process at University of Helsinki. The application process, as you already learned, includes a mandatory sales pitch to Business Finland. That will be about 1 month before the application deadline. That will be sort of your entrance ticket to the whole application process, because you will need to get either a yellow or a green light from Business Finland in order to actually even be eligible to apply. Or let's say they will encourage you to apply. The innovation units at different universities coordinate these application types, and in addition to the coordination, Helsinki Innovation Services, which is essentially the innovation unit at Helsinki University, also supports the applicants. I'm quite sure that all of you are very experienced in writing grant applications, but I think especially Philip pointed out that this requires a bit different mindset, as this is commercialization funding, and therefore we have seen that it's valuable to have a short preparation process for this. The preparation process at our university is essentially the so-called NABC training program. The letters come from need, approach, benefit, and competition. This is the format that Business Finland encourages you to use when pitching and preparing your project plan. So the training will consist of 3 afternoon events. In the current state, held via Zoom, and then an internal university training pitch. Also, the afternoon sessions will include pitch training. So in addition to the pitch training, we'll offer you project plan and application advice and small hands-on workshops. Business Finland will evaluate certain aspects of your application. They will be interested in the scalability of your business, the commercial skills in your team, the business model you are planning. So there are certain topics that probably will benefit from brainstorming and sparring together in these sessions. You're very welcome to register to this NABC training program. Latest 18th of December. The link for the application you will find either via the HIS or the helsinki.fi webpage, and my colleague also copied the link to the chat box. You can reach us via our webpage, you can email me. You'll also find us on Twitter and LinkedIn. So please feel free to be in touch with us if you have any questions about this funding type. Or if you have questions already now, I'm happy to take them, of course.
Thank you, Kaisa.